Hello, my dear students of class 12 science. Welcome to biology class. We are dealing with the chapter biotechnology, principles and processes, and we are in the topic process of recombinant DNA technology. Okay, so till now we have dealt with the few process or steps. The first one was isolation of genetic material or DNA. Second one was separation of isolated DNA fragment, which was done by the help of aerosol electrophoresis. Third one is cutting segment of isolation of DNA fragments by the help of restriction endonuclease enzyme. And fourth one was amplification or multiplication of gene of interest by polymerase chain reaction or PCR. So till there we have learned in our previous classes. Okay, so today we will begin with the next step or the next process of recombinant DNA technology, that is insertion and ligation of DNA segment into vector and formation of recombinant DNA. Okay, so all of you are aware with this process, because once we used to identify the foreign DNA having the gene of interest, we have to cut it by the help of restriction endonuclease enzyme. Clear? Then for insertion of that foreign DNA having the gene of interest, we require a vector DNA. Generally we used to use the plasmid vector PBR3 to try to recall the structure of that vector. Clear? So first of all, we will isolate the gene of interest from the foreign DNA having the sticky ends. So we will cut it with the help of restriction endonuclease enzyme, and we will insert that foreign DNA having the gene of interest into the vector DNA. Remember, both the vector DNA and the foreign DNA needs to be cut by the same restriction endonuclease enzyme. Okay, so ligation or the insertion of foreign DNA having the gene of interest into the vector DNA is possible by the help of the discovery of molecular glue, which is called DNA ligase. Okay, because the enzyme DNA ligase will help in establishing the phosphodiester bond between the two different DNA, which is also called splicing, and this will lead to the formation of recombinant DNA. So now the recombinant DNA has been produced. So after the formation of recombinant DNA, we have to go to the next step or the next process, that is insertion of recombinant DNA into the host cells. Clear? So once the recombinant DNA has been created, it has to be inserted into the host cells. Generally we use the E. coli cells or the Escherichia coli cells. Okay, so recombinant DNA is introduced into the host bacterial cells after it is made competent, because the host DNA or the host bacterial cells will not be able to take up the foreign DNA or the recombinant DNA easily. They have to be made competent so that it will be able to take up the recombinant DNA. Okay, so in the last class we have learned about the process by which we can make the bacterial cell competent to take up the foreign DNA or the recombinant DNA. Clear? So over here, for insertion of recombinant DNA into the host cells needs to be made competent so that they will be able to take the recombinant DNA. So once the host cells or the bacterial cells will take up the recombinant DNA, they will become transformed. And there are several methods by which we can introduce the recombinant DNA into the recipient cell or into the host cell. So let's go through the method by which we are able to introduce the recombinant DNA into the recipient cell or into the host cells. The first one is the gene gun method. Okay, the first one is the gene gun method, which is also called the biolistic method. Okay, so in this method we will mainly use to transfer the recombinant DNA in plant cells. Remember, so gene gun method is mainly employed in the transfer of recombinant DNA in plant cells. So over here in this method, the microparticles of gold or tungstens are coated with recombinant DNA and they are bombarded with high velocity into the plant cell walls. So when we used to bombard the recombinant DNA with high velocity into the plant cell wall, the recombinant DNA gets incorporated into the host DNA. Second one is micro injection. In this process, we will use the microsyringe or the glass micropipette for the transfer of recombinant DNA into the animal cells. Okay, so here in this method, we will directly inject the recombinant DNA into the nucleus of the animal cells by the help of microsyringe or the glass micropipette using two micromanipulators. So this technique was mainly used in animal cells, but nowadays it's also been used to transfer the recombinant DNA in plant cells also. The next method is transformation, in which the host cells will take up the foreign DNA from its surroundings. And in this case, the recipient bacterial cells are made competent to take up the recombinant DNA by placing them in cold and dilute calcium chloride solutions. Okay, so by this way they will be competent and they will be able to take up the recombinant DNA. Next one is transfection. In this process, the introduction of recombinant DNA is done by the help of phage particles which infect bacterial cells. Phage particles means bacteriophage, the virus which can infect the bacteria. So by the help of the phage particles, we are able to transfer the recombinant DNA into the bacterial cells in the process of transfection. So my dear, remember that phage particles means the bacteriophage, the viruses which can infect the bacterial cells. Last one is electroporation method, in which the introduction of recombinant DNA is done by the help of the high voltage electric pulse. Clear? So the bacterial cells are exposed to a very high voltage of electric pulses. By doing this, so a pore will be created in the cell membrane of the bacterial cells and the plant cells, and by the help of these pores, the recombinant DNA can enter into the host cells. So these are the methods by which we can introduce the foreign DNA in the host cells. Remember, by gene gun method or by ballistic method, microinjections, transformation, transfection and electroporation. So these are the few methods by which we are able to introduce the foreign DNA in the host cells. Clear? Now, next step is selection of transformed cells. Now, what does transformed cell means? Try to recall that in case of recombinant DNA technology, we are creating the recombinant DNA having the gene of interest. Okay, and after creating that recombinant DNA, we are inserting it into the host cells. Let's say, for example, Escherichia coli or E. coli. Now, how to identify whether that cells has been transformed or not? So for selection of transformed cells, we have to take the help of selectable markers and try to recall the structure of the vector PBR322. We have got two selectable markers. One is AMPR and the second one is TETR. All these things, all this process we have discussed already in our previous classes. So try to recall it back. Okay, so AMPR means ampicillin resistant site and TETR means tetracycline resistant site. If a particular cell contains both AMPR and TETR, then it will be resistant to the antibiotic ampicillin and tetracycline. So kindly go through our previous classes regarding the selection of transformed cells and the non-transformed cells, or we used to call it recombinant cells and non-recombinant cells. Okay, so kindly go through previous classes of the process of selection of transformed cells and non-transformed cells. So 7th step is the selection of transformed cells. It will be followed by the last step, that is culturing the transformed host cells, culturing the transformed cells for cloning of recombinant DNA. So here we have to go for the gene cloning. So what we are doing is that we have created the recombinant DNA, we have inserted into the host cells, into the host DNA. Once the recombinant DNA enters the host cells, let's say E. coli cells, that recombinant DNA can replicate, can multiply independently within the body of host cells. and when they used to replicate independently within the body of host cells, they will produce numerous copies of recombinant DNA, and this process is called gene cloning. And the cloned recombinant DNA produced in abundance, in higher number in a bacterial culture, will be isolated, it will be extracted, it will be purified, and it will be analyzed. Now it has been extracted, it has been purified, it has been analyzed, and now it will be used for the culturing transformed cells for obtaining desired gene product. Clear? So in this part, what we do, the recombinant DNA which has been created or which has been formed in abundance in higher number within the host cells has been extracted, has been analyzed, has been purified. Now that recombinant DNA will be utilized to obtain the desirable product. Whatever the product we want to make, we can obtain it by using that recombinant DNA. Okay, so for this one, the cloned DNA, whenever they are introduced into the host cells, they will be expressed. Okay, that DNA will lead to the formation of mRNA, and mRNA will lead to the formation of protein. Clear? So that means from DNA, from recombinant DNA, there is formation of messenger RNA, and from messenger RNA there is formation of proteins. So therefore the process or the protein that has been expressed or that has been formed in the heterologous host is called the recombinant protein. Clear? So a mammalian gene can express in a bacterial cell only if it is accompanied by proper regulatory regions. Also, the gene should not contain introns because bacterial cells do not have enzyme for processing primary transcript of mRNA. So we will go in details in the chapter molecular biology about intron and exon. Okay, and you all are aware about the topic reverse transcriptase, where there will be formation of what? There will be formation of DNA from mRNA. Clear? So in the chapter molecular biology, we will go in details about exons and introns. So here we have finished the process of recombinant DNA technology, and finally we have obtained the desirable product by creating the recombinant DNA. Now next one, let's talk about the growth and maintenance of cell culture and biochemical engineering process. Okay, sorry. So here we will try to produce the product in a higher number, in a higher amount. Okay, so therefore here we'll take the help of bioreactors or the fermenters for the large scale productions of bioengineered products or recombinant proteins. So in order to obtain the product at the higher number or in large quantities, we will take the help of bioreactors or the fermenters. Now what are bioreactors? So bioreactors are the large vessels. So you can see the figure over here. These are the two types of bioreactors: a simple stirred tank bioreactor and sparse stirred tank bioreactor. But we have to only go through the simple stirred tank bioreactor. This is very, very important for your examinations. Okay, so we'll just go through the simple stirred tank bioreactor. So what is bioreactor? So bioreactors are large vessels in which raw materials are biologically converted into a specific product using large volumes of culture of microbes or plants, animals, or the human cells. So bioreactors are mainly used to obtain the larger productions of the product from the recombinant DNA, where we use the larger volume of cultures of the microbes or the plants, animals, or human cells. Okay, so these have adequate arrangement for aerations. There will be proper space for the air. There will be proper process for agitations to mix the materials. There will be drain or overflow vent to remove the waste biomass along with their products. So optimum growth conditions like temperature, pH, oxygen, vitamins, nutrients, and substrate, these are all maintained within the bioreactor. Yeah. So let's go through the stirred tank bioreactor. So each reactor is a large closed cylinder vessels made of steel. It has a curved base to facilitate the mixing of culture broth. So it has the following features. So let's see. It has got the culture broth. You can see the stirred tank bioreactor. It has got the culture broth. Number 2, a stirrer plate or stirrer blades magnetically coupled to the shaft for stirring the broth. So when we see over here, we can see the structure of a bioreactor or stir tank bioreactor, which are large vessels which is mainly used to obtain the product as a larger amount. Clear? Number 3, we can see the oxygen delivery system for the air inlet, from where the air can enter inside the bioreactor. A foam control system is there. We have got temperature, pH control systems, and a cooling jacket, and sampling pots for removing the small samples of culture from time to time. These are the few features of the stir tank bioreactor. So you can see over here, we have got the motor, we have got the foam beaker control over here, we have got the flat bladed impeller for mixing the broth. Okay, we have got the cooling jacket which can maintain the temperature. We have got the sterile air which can generally come in and out of the motor. Okay, and we have got the steam for sterilization. Then we have got the nutrient line, inoculum line, and we have got the pH control. So these are the few features of stir tank bioreactors which are mainly used to obtain the product at a larger amount. Clear? Okay, so just go through the stir tank bioreactor. No need to go through the space stir tank bioreactor. Now next topic is the downstream processing. Okay, because we have created the product by using the bioreactors in a larger amount. So after creating the product, we have to go through the downstream processing. So the recovery, purification of biosynthetic product from the growth medium is called downstream processing. So once the product has been produced by the help of recombinant technology in a stir tank bioreactor, then we have to go for its recovery, then we have to purify it. So this process of recovery of the product followed by its purifications is known as downstream processing. So what are the different steps in downstream processing? So it includes filtration, centrifugation, sedimentations, and various types of chromatographic techniques and electrophoresis. Okay, and after the product has been purified, it has to go for clinical trials, like in case of drugs. Okay, whether those product has got any side effects or not. So downstream processing and strict quality control are essential features before the product is being used for the or is being marketed for the human use. So once the product has been produced, it has to go for purifications, which is called downstream processing, and after its purification, it has to undergo clinical trials whether that product has any side effect or not. Okay, only after clinical trials, if it is safe for the human use, then it can go for marketing. Okay, next one is the application of recombinant DNA technology. Okay, so recombinant DNA technology has opened up a wide opportunities for the mankind in various fields, such like in case of medicines, we can manufacture insulin. Okay, we can create vaccines, even we can create the growth hormones. Then we can diagnose the different types of diseases. We can go for gene therapy. It's also used in agriculture for the production of genetically modified crops, and it's also used for environmental improvement and in case of industrial fields. So this is the application of recombinant DNA technology. So here we have finished the chapter biotechnology, principles and processes. In the next class, we'll begin a new chapter, that is biotechnology and its application. Okay, so this much for today. Thank you very much. See you in next class with a new chapter, that is biotechnology and its application. Thank you.